Let's take a closer look at what happens to the DNA during the cell cycle. First of all, during interphase, the DNA is uncoiled inside the nucleus. A copy of the DNA is made, and it had to be uncoiled so that you could get to the DNA to copy it. If it's all wrapped up tightly into a chromosome, it can't be copied. This uncoiled DNA is known as chromatin. And if you look under a microscope, you won't be able to see it. The little filaments of DNA are so thin that they're invisible. Next, during mitosis, the DNA coils up to become chromosomes. So let's look at what happens during the phases of mitosis. During prophase and metaphase, the DNA is coiled up like a slinky. It just twists and twists and twists. This way, it's all wrapped up into a nice package to where a little piece of it won't break off and get lost when it's being separated and divvied up between the two new cells. The DNA exists as two identical sister chromatids. The cell has made a copy of the chromosome, and the two identical copies are called sister chromatids. Make sure you draw an arrow to each of these. Then, together, this is all considered to be one chromosome. The two sister chromatids in the chromosome are held together by a centromere. And the DNA is visible because you can see the chromosomes because they will look dark. During anaphase and telophase, the two copies of the DNA are separated and pulled into what become two new cells. So the DNA is still coiled into chromosomes, but the sister chromatids separate. So here's one chromatid left. And go figure, when you have a pair of sister chromatids, that's considered a chromosome. And when you have a single sister chromatid, that's considered one chromosome. See, you count centromeres, not chromatids. So during anaphase and telophase, the DNA is still coiled up. The pairs of sister chromatids separate, and they are pulled apart by the spindle fibers. The spindle fibers look like little strings. They are made by the centrioles, and the centrioles use those spindle fibers to pull the chromatids apart. Each sister chromatid is now a single chromosome, and the DNA is still visible under the microscope because it's still coiled up into a chromosome. So yes, a pair of sister chromatids is called a chromosome, and a single chromatid is still called a chromosome. Then the cell goes back into interphase, the DNA uncoils, and then the DNA can be used as the cell makes new parts and new organelles, makes proteins, and just continues on living. Then, when it's ready to divide again, it will make another copy of the DNA, continue to make some last-minute preparations, and then once again enter mitosis.